our next step of the Krebs cycle. So till now, we've gone on to make our succinyl CoA, which was our four-carbon compound. From this four-carbon compound, we're going to now make another four-carbon compound. So we're just going to make a little, just going to tweak a little bit of the changes and do nothing very, very special with the succinyl CoA. And from the succinyl CoA, we'll just make the succinate, right? Which both of them are four-carbon compounds. There's not a lot that is happening here, except for the fact that we're now producing GTP, right? So from this succinyl CoA, we've made the succinate compound, right? So I think we've covered this step in the previous video, but let's just go over it again. So succinyl CoA was being converted into succinate by the enzyme succinyl CoA synthetase, right? And it produces GTP, which is equivalent to ATP. So one GTP is equal to one ATP, right? It's the only substrate level phosphorylation of the TCA cycle. Whenever there's a vitamin B6 deficiency, vitamin B6 is needed for the ALA synthase enzyme. When there's an ALA synthase enzyme which is impaired, there's less heme production. Why? Because the succinyl CoA is converted into the ALA enzyme, and this is the first step in heme synthesis. So heme synthesis will be impaired if there's a vitamin B6 deficiency. Also, lead inhibits heme synthesis at two steps. Lead inhibits heme synthesis at the ALA dehydratase step and the pherochelatase step. So there's less heme production, and the patient is susceptible to getting anemia. Okay, now we've already covered this step in the previous video at the end. We will now take the succinate four-carbon compound and convert it into the fumarate four-carbon compound. And for that to happen, we need to understand our step 6 here. So step 6 is where you take the succinate and you convert it into fumarate using the succinate dehydrogenase enzyme. This produces the FADH2 molecule. It's a very high energy electron carrier. A unique fact to mention here is that this is the same enzyme. The succinate dehydrogenase is the same enzyme that is being used in the complex 2 of the electron transport cycle, the electron transport chain that we will do after completing the Krebs cycle. So remember that the succinate dehydrogenase enzyme is common to the TCA cycle and common to the electron transport chain. And this is the only TCA enzyme, this is the only TCA enzyme that is embedded in the inner mitochondrial membrane. So the electron transport chain entirely happens in the inner mitochondrial membrane, right? So this is the only enzyme of the TCA cycle that is embedded in the inner mitochondrial membrane. Another thing I would like to mention here is that there's also a mutation that we've seen with the succinate dehydrogenase enzyme. So whenever there's a mutation of the succinate dehydrogenase enzyme, it leads to tumor suppression mutation, right? So it's associated with like tumors like preganglioma, pheochromocytoma, gastrointestinal stromal tumors, in some cases, renal cell carcinoma, some hereditary syndromes. So succinate accumulates, right? It acts as an oncometabolite. Um, and this is similar to the IDH mutation we just saw in the previous video. And when succinate accumulates and acts as an oncometabolite, it stabilizes the hypoxia inducible factor, and that promotes angiogenesis and tumor growth, right? Okay, moving on. The causes, I told you, the cancers that are most susceptible when we get succinate dehydrogenase mutation. We have paragangliomas, pheochromocytomas, wild type gastrointestinal stromal tumors, renal cell carcinoma, in some rare hereditary syndromes. And the mechanism is basically that succinate accumulates and pseudohypoxia signaling is activated. So it stabilizes hypoxia inducible factor, and that leads to cancer. A USMLE clue could be young patients with multiple paragangliomas with an SDH mutation. Remember, succinate dehydrogenase connects the TCA cycle to the electron transport chain. All right, so now at this point, you have converted the succinate into fumarate, right? You've used the succinate dehydrogenase enzyme and you've produced a molecule of FADH2. We now use the fumarate molecule to convert it into the malate, right? We're almost on the second last step of the Krebs cycle. So we now will use the fumarate, which was our four-carbon compound, and convert it into the malate. This was using our enzyme fumarase. So can you see that fumarase takes the fumarate compound and converts it into malate? Water is added in this step, and the enzyme that we use during this happening is the fumarase. The clinical correlation is basically a fumarase deficiency. Whenever we have a fumarase deficiency, by the way, this is rare, it's autosomal recessive. And fumarate deficiency is rare, it's autosomal recessive, it has some causes. What causes basically fumarate deficiency? There will be a severe developmental delay, some kind of a hypotonia. Seizures will be seen, encephalopathy will pursue, poor bone growth, and brain abnormalities. All of these are causes of basically the fumarate deficiency. A FH mutation can also persist. An FH mutation is the mutation of fumarate hydratase enzyme. Causes include hereditary leiomyomatosis and renal cell carcinoma. Hereditary leiomyomatosis and renal cell carcinoma, you can remember it with this mnemonic HLRCC. Fumarate accumulation is also a non-cometabolite. When fumarate accumulates, it persists into cancers, right? So remember, whenever we have some kind of a mutation, and this basically all these TCA cycle accumulations lead to tumor growth. They become cancerous, right? Okay, so this is our basically succinate being converted to fumarate, and now we need to convert the fumarate into the malate, right? And malate was done. We converted the fumarate into malate using the fumarase enzyme, and the FH mutation was leading to a fumarate accumulation, which was becoming a non-cometabolite and persisting into a cancer. Now, the last step is converting the malate and regenerating the oxaloacetate from the malate so it can grab onto another acetyl CoA and repeat the Krebs cycle. So, malate basically, look at this step 8 here. The step 8 is basically taking the malate and converting it into oxaloacetate. This is done by malate dehydrogenase enzyme, right? The reaction basically produces an NADH molecule, right? So oxaloacetate is regenerated, the cycle repeats, and an NADH is produced, which is a high energy carrier molecule, right? A clinical correlation of this would be malate is involved in the malate aspartate shuttle. This is important for transferring the cytosolic NADH into mitochondria. So the malate also participates in this shuttle. It's a malate aspartate shuttle. This transfers the cytosolic NADH, reducing equivalents into the mitochondria, and it allows these electrons to enter the electron transport chain efficiently, right? So you have, can you look at the cytosol here? So this is the cytosol of the cell, and this is the mitochondrial matrix. What the malate does is what the malate basically does is it grabs onto this NADH, right, and it throws it into the mitochondrial matrix using the malate aspartate shuttle. And this allows efficiently the electrons to enter the electron transport chain inside the mitochondrial matrix. This step completes the TCA cycle by regenerating the oxaloacetate, which is ready to accept the next acetyl CoA enzyme. Right, so that's something about the Krebs cycle. If I show you a little bit of a summary here, on the Krebs cycle, what we will notice is where, first of all, the acetyl CoA was coming, right? It was a two carbon compound. It was being added to the oxaloacetate, and using the citrate synthase, it was being converted into the citrate enzyme. No energy was produced in this step. Then the citrate was being converted into isocitrate using the aconitase pathway, and you were basically creating the isocitrate here. Then we were using the isocitrate compound to make the alpha ketoglutarate compound, and during this you were producing one NADH and one carbon dioxide. This was your oxidative decarboxylation, and you were using the isocitrate citrate dehydrogenase enzyme. Then you were converting the alpha ketoglutarate into succinyl CoA. During this process, you also created one carbon dioxide molecule, you created one NADH molecule, and removed the CoASH group. This is the alpha ketoglutarate dehydrogenase complex that was doing this, and this is very similar to our pyruvate dehydrogenase complex. It also requires the five cofactors that we studied, right? And then we have basically the we have the succinyl CoA. The succinyl CoA basically then gets converted into succinate, right? And during this, you are creating one GTP, and this was done by succinyl CoA synthetase. You created one GTP, which is equivalent to one ATP, and then you were using the succinate in compound to convert it into fumarate, and this was done by the succinate dehydrogenase. This is common to the electron transport chain and the Krebs cycle. The one molecule of FADH2 was produced as a high energy molecule, high energy carrier molecule, and then you were using the fumarase. The fumarase was being used to convert the fumarate into the malate, and the malate aspartate shuttle was helping in sending this NADH down the electron transport chain, right? And then the malate was being used to regenerate the oxaloacetate using the malate dehydrogenase enzyme. and that's how we regenerate the oxaloacetate, ready to grab another acetyl CoA molecule. There's a high yield USMLE regulation here. This entire pathway is activated by a low energy state, which is shown by high levels of ADP and calcium. Calcium, because the exercising muscles releases calcium, and all of this cycle is basically inhibited by high levels of energy states, which are represented by high levels of ATP, NADH, citrate, and succinyl CoA. All of these stop the Krebs cycle from happening a lot, because all of these represent a high energy state in the cells. The three major regulatory enzymes that I would point out for the Krebs cycle are the citrate synthase here, and then we have the isocitrate dehydrogenase here, and the alpha ketoglutarate dehydrogenase complex, which is most similar to the pyruvate dehydrogenase complex, and they have the same cofactors, right? The most important clinical correlates to memorize here would be like the alpha ketoglutarate dehydrogenase complex would be inefficient in a vitamin B1 deficiency, because thiamine pyrophosphate is needed as a cofactor here. Arsenic poisoning would also inhibit the alpha ketoglutarate poisoning, because it binds onto the lipic acid, which is one of the cofactors needed for the alpha ketoglutarate dehydrogenase complex. So in arsenic poisoning, there will be a garlic breath in the patient. In vitamin B1 deficiency, thiamine deficiency, obviously, there will be lactic acidosis, neurological dysfunction. There's sometimes known as, we sometimes see the IDH mutation, right? We saw the IDH, the isocitrate dehydrogenase mutation. This became, when there was a mutation here, we had let's just go back and look at this here. So when there was a IDH mutation, what was the problem? We were producing too much 2-hydroxyglutarate instead of alpha ketoglutarate, and that was becoming an oncometabolite. It was participating in cancers, right? And then so basically 2-hydroxyglutarate was being accumulated. It was participating in cancers such as glioma and AML. And then we have the succate dehydrogenase mutation. The succate dehydrogenase was common to the electron transport chain and the Krebs cycle. The succate dehydrogenase mutation was part of the complex 2 of the electron transport chain also. And by the way, if there's a succate dehydrogenase mutation, again, you have an oncometabolite and cancer persistence of paragangliomas, pheochromocytoma, because of the because of the stabilization of the hypoxia inducible factor activation. Then the fumarase deficiency was also leading to an oncometabolite forming. Fumarase deficiency was leading to developmental delay and seizures. And the FH mutation, the fumarate hydratase deficiency, I told you was I just said it was related to the cancers. Hereditary leiomyomatosis and renal cell carcinoma is very common with fumarase hydratase deficiency mutation. It's called FH mutation. FH mutation. Then we had the fluoroacetate poisoning, which was basically, fluoroacetate poisoning was inhibiting the aconitase. This aconitase was inhibited by fluoroacetate poisoning, and this basically inhibits the TCA cycle inhibition, right? So that's a little bit of a review of the Krebs cycle. I hope I got all the things right. If I made any mistake, I'm very sorry. Please drop down in the comments if there's anything I said wrong. I would want the other person to learn from it, everybody to learn from this. Thank you.